For this week's lecture, we're going to be talking about the fundamentals of the nervous system. Essentially, we'll be talking about the cells that make up the nervous system as well as the individual components of the nervous system. This should set the stage for us to be able to talk about some more in-depth functional aspects of how the nervous system functions later on in the semester. So let's get started. So neurons are the fundamental units of the nervous system, and I have to say that they are the most beautiful cells in the entire human body. On the right here are examples of two neurons. The one on the top has been stained for particular proteins that fill the cell up, so the neuron is actually in green and blue. And then the little red particles and dots on the extensions of the neurons are actually other neurons making contact with this one. And we'll talk about the ways in which neurons communicate with each other in the coming lectures. The neuron's complex connections allow for specialized areas and functional neural networks. It's the ability for neurons to communicate with one another in sequence or in parallel determine the effectiveness of particular functions that the nervous system oversees. So about 150 years ago, the perspective of what the nervous system was built of was very different than what it is today. So Camillo Golgi was an Italian physiologist who came up with a stain that allowed physiologists to see whole neurons under the microscope, and they look like this bottom picture on the right. And based on his observations under the microscope, he believed that the nervous system was one continuous network, that it was like one big net of information that kind of got activated in coordination to control different functions that he called the nerve net theory. Around the same time, Santiago Ramon y Cajal came up with the neuron doctrine. So he used Camillo Golgi's stain, which is called the Golgi stain and is still used today. And he, in his observations, drew meticulously beautiful neurons. And based on his drawings of many different brain areas, he concluded that neurons were actually the individual units. They were the smallest, but the individual functioning unit within the nervous system. And so he disagreed with Camillo Golgi, but they both ended up sharing the Nobel Prize in Physiology and Medicine in 1906. Even though the neuron doctrine still is the fundamental tenet of neuroscience to this day, Camillo Golgi stood by his nerve net theory. He was a very, very stubborn neuroscientist. At first glance, looking at a neuron, you see that it actually has very specific distinguishing parts. It looks nothing like, for example, a liver cell or a muscle cell or, a skin cell. It's got very distinctive areas, and these distinctive areas perform different functions within the neuron. So the most prominent structure in the neuron is the cell body, or soma. It ranges in size from 5 to 100 micrometers in diameter and contains all of the microstructures of the cell. So we'll be talking about these microstructures or organelles in a couple of slides. There are two major groups of projections from the neuron. They both originate from the cell body. One of them is the dendrite, and the other one is the axon. This entire thing is the axon. So the dendrites serve as the receiving portion of the neuron and accounts for the vast majority of its surface area. They branch and branch, working to establish networks with other neurons. So it's at the point of the dendrites that other neurons communicate with one another. The axon is the sending portion of the neuron and carries the nerve impulse down the axon, but it starts at an area of the axon known as the axon hillock. That's like the head or the beginning of the axon. And the nerve impulse is electricity, essentially, generated in the axon that's carrying the message to the end. Because the signal is electrical, the axon is covered with fatty insulating material called, I lost my thing again, called myelin sheath. The myelin sheath serves to insulate the nerve impulse, allowing it to move efficiently down the length of the axon. However, there are regular spaces between the myelin to help regenerate the nerve impulse, and we'll discuss how it does that in the next few weeks. But these spaces are known as nodes of Ranvier. The axon ends by branching with little buttons at the end of it. These little buttons house the chemical messenger that neurons use to communicate with one another. The chemical messenger is called a neurotransmitter. Within the cell body contain all of the internal components of a neuron, and you might notice, if you've taken biology at some point, that many of the cell body structures of a neuron are similar to those found in many other cell bodies. There's the nucleus, which we've discussed, that contains all of the cell's genetic material, such as the chromosomes. The cell membrane is what keeps the inside of the cell separate from the outside. We'll be discussing the parts of the central cell membrane in the coming weeks when we talk about what makes the neuron unique enough to be able to generate the nerve impulse. Right next to the nucleus is a structure called the rough endoplasmic reticulum. Together with the nucleus, it participates in protein production. So remember we talked about the mRNA last week? The mRNA from the nucleus that's generated by using the DNA enters the rough endoplasmic reticulum, and the ribosomes that give the rough endoplasmic reticulum its rough, studded appearance are the ones that make the protein from the mRNA by reading it. So the ribosome, as I just mentioned, is the primary site of protein synthesis. We also have the smooth endoplasmic reticulum, separate from the rough because it has a smooth outer membrane. It's responsible for producing things like fats, lipids, or steroid. The mitochondria is really, really important in the neuron. It is the sole producer of energy that a neuron uses. The Golgi apparatus is also another structure that's important in a neuron. Because neurons communicate with one another using chemical messengers, these chemical messengers often get packaged into the Golgi apparatus, vesicles we call them, before they're released. You also have proteins, such as the cytoskeletal proteins, that work to give the neuron its shape. So they work like the skeleton of our body, and they consist of scaffolding to allow for the transport of materials or change the shape of the neuron based on what is needed. Lysosomes are the cell's digestive system. They're responsible for breaking down proteins and lipids. So as we need it, we'll be talking about some of these organelles in greater detail as they come up, but for now, this is all you need to know. Okay, let's talk about the types of neurons that we can find in the nervous system. There are essentially 3 different types of neurons if we classify them based on their extensions. So neurons can either be multipolar, bipolar, or unipolar. Multipolar neurons have one axon originating from the cell body and many, many dendrites exiting from the cell body as well. That's what makes them multipolar. Bipolar has one dendrite extension that branches and one axon that leaves the cell body. Unipolar is a bizarre-looking alien-like neuron. It has the cell body sitting outside of the extension of the dendrite, kind of like a head, and typically what you would see is bipolar and unipolar neurons in sense organs, such as the eyes, the skin, nose, and ear and mouth. The vast majority of the neurons that exist in the nervous system are multipolar in structure. Besides neurons, glial cells make up another important group of cells within the nervous system. So there are many different kinds of glial cells. Astrocytes are one of them. They've got the star-like appearance, which is why they're called astrocytes. They have many, many different functions. Some of them are listed up here. They fill the spaces between neurons. They influence whether extensions from neurons grow or retract. They regulate the chemical content of the brain, and they engage in something known as phagocytosis, which is essentially like Pac-Man, gobbling up any debris that might exist within the brain if there's any damage or kind of trash. There are two groups of myelinating glia. So remember that fatty insulation that surrounds the axon that I talked about earlier? Well, they're made by two different types of cells. The myelin can be produced by oligodendrocytes or Schwann cells. They both produce myelin, and they produce spaces within the axon known as the nodes of Ranvier that I mentioned earlier, but only one of them is found in the central nervous system and the other is found in the peripheral nervous system. Again, these two concepts I will introduce in part 2. These two concepts I will introduce later in the lecture. But oligodendrocytes are the ones that are typically found in the central nervous system, and Schwann cells are the ones that are found in the peripheral nervous system. So just to give you an idea of what some of these cells look like, the astrocytes are here, and we'll be talking about microglia in a second. Here is an example of an oligodendrocyte, and this is the way in which it wraps around the axon. It's kind of like a paper towel roll. The central portion, the brown portion of the paper towel, is the axon, and then the actual paper towel itself is what could be the myelin surrounding the axon. So here's another better picture of myelinating glia. Oligodendrocytes are the ones that are typically found in the central nervous system, and they can myelinate multiple axons at any given time. So they can, because of the branches that are coming off of them, they often myelinate, anywhere from 4 to 6 axons. Schwann cells, on the other hand, which are only found in the peripheral nervous system, typically myelinate one axon alone because the entire cell has to wrap around to create the myelin. What's interesting is that when there's dysfunction of the myelin, because we rely so heavily on the myelin to conduct the nerve impulse, whenever there's damage to the myelin, that ends up weakening the nerve impulse, resulting in diseases like multiple sclerosis. So multiple sclerosis is a disease where the body's immune system essentially attacks the myelin. No one has any idea why, but it attacks the myelin and causes scar tissue to develop, preventing the nerve impulse from effectively being transmitted down the length of the axon. But because, again, we rely so heavily on myelin to conduct the nerve impulse, any damage to it can ultimately affect nervous system function in human beings. So the last kind of group of glial cells that we're going to talk about are radioglia and microglia. So as I mentioned to you earlier, here's a microglia. Microglia serves the immune system of the brain. The brain has a specialized protection that we'll talk about in the next slide called the blood-brain barrier. So it doesn't allow for antibodies, which the immune system of the body makes, to enter the brain. So there's really no protection for the brain against invading microorganisms, but microglia serves that particular function. Whenever they're invading microorganisms, microglia kind of take up the charge and end up fighting off those organisms. Radioglia are really, really cool. They only exist in the brain during development, and their sole purpose is to serve as kind of train tracks for newly born brain cells to their final destination. Following development, radioglia actually turn into astrocytes. So they're really, really neat. If you want to learn more about it, let me know. I'm happy to talk to you more about it. All right, as promised, we're going to be discussing the blood-brain barrier. So the brain is critical in terms of function. Any disruption to its delicate balance of chemicals can really wreak havoc. And so there exists this protection, this physical protection known as the blood-brain barrier, to keep substances that might enter the body away from the brain that may ultimately disrupt its function. So there are two factors that contribute to the blood-brain barrier. The first is the blood vessels. Unlike the rest of the body, there are absolutely no gaps in the cells that make up the capillary or blood vessels in the brain. So the only way substances can get in and out of the blood vessels is through active transport. They have to be physically pumped out of the blood vessel, or they just have to diffuse. They have to pass through the membranes of the cells. So substances can't get through easily through the blood vessels within the brain. The other one is the astrocyte end feet. So astrocytes have these suction-like protrusions that rest up against the blood vessels of the brain, and they serve as a physical barrier between neurons and the actual blood vessel wall. Astrocytes also help transport nutrients. So as I mentioned earlier, the only way substances can get from the blood vessels to the brain is through active transport or pumping, and astrocytes are one of the things that help pump nutrients like glucose into the brain from the blood vessels. What's interesting is that not all areas of the brain have a blood-brain barrier, and there's a reason for this. Area postrema, which is found in the brain stem, like the primordial region of the brain, has a weak blood-brain barrier. This particular brain area is also known as the vomiting center of the brain. It needs to have a weak blood-brain barrier so it can detect chemicals that could ultimately be harmful for our body and induce the vomiting reflex. And so not all areas have a very tight blood-brain barrier. The area postrema is one of them. So let's kind of shift gears a little bit and talk about neuroanatomy, or how the brain is structured. So the major divisions of the brain are kind of diagrammed here. Two major divisions of the brain are the central nervous system that I talked about earlier and the peripheral nervous system. The central nervous system can be further subdivided into the brain and spinal cord. All other components of the nervous system fall into the peripheral nervous system category. But the peripheral nervous can be functionally subdivided into the somatic and autonomic nervous system, and the autonomic nervous system can be further subdivided into the sympathetic and parasympathetic nervous systems. And we're going to talk about all of these in a few minutes. But first, there's a clear difference between the peripheral nervous system and central nervous system in terms of recovery from injury. When you cut a nerve in the peripheral nervous system, it's able to regenerate and regrow. This is in contrast to injuring the central nervous system. If you break axons in the nervous system or have an axon tear in the nervous system, there's absolutely no regrowth or recovery. That's why concussions and traumatic brain injury are so damaging, because there's absolutely no way to recover the axons that might have been damaged or cells that might have been damaged in the central nervous system. This is in contrast to, you might have a cut in your arm and lose feeling, but in a few months, these axons are able to regrow in the peripheral nervous system, and you will regain feeling again. So in terms of function, they're very different, but the capability of regenerating from injury is also very different. The peripheral nervous system can recover. The central nervous system, unfortunately, cannot. So let's talk about nerves first for a second before we talk about the peripheral nervous system. So we've discussed axons, right? Axons can be bundled together to form a nerve. So when we talk about nerves, they're actually large bundles of axons that are originating from the central nervous system, either the brainstem or the spinal cord. So what you have here on the right are nerves that are entering and exiting the spinal cord. Most nerves are made up of two different kinds of nerve fibers. They can either be sensory, they're responsible for bringing information from the body to the central nervous system, or they're motor. In fact, they control the muscles of the body. So most spinal nerves are what are known as mixed. They have both sensory and motor capability. Another way to think about this is the nerves are carrying efferent and afferent extensions. Efferent extensions are those that are carrying information from the central nervous system to the rest of the body, and they tend to be motor in nature. And afferent are the ones that are carrying information from the body to the central nervous system, and they're often sensory in nature. So the peripheral nervous system is pretty much any part of the nervous system that's not covered in bone. So as I mentioned, the central nervous system is the brain and spinal cord. Those two structures tend to be covered in bone, the skull or the vertebral column, but any other portion of the nervous system is considered the peripheral nervous system. Structurally, it can be divided into two parts. You have the cranial nerves that originate from the brainstem and the brain, and then you have the spinal nerves that originate from the spinal cord. But functionally, as I mentioned earlier, they're divided into the somatic and the autonomic nervous systems. So the cranial nerves all originate from the brainstem or the base of the brain, and they control a vast variety of different organ systems in the face and body. The spinal nerves originate from the spinal cord, and they're shown here. They're divided into the cervical nerves, thoracic, lumbar, and sacral nerves. So the somatic nervous system includes both cranial and spinal nerves. The somatic nervous system can be thought of as the voluntary portion of the peripheral nervous system. It gathers sensory information from the sense organs, but also controls movement of the skeletal muscles. The autonomic nervous system, or the involuntary portion of the nervous system, regulates things like digestion, cardiac muscles, and gland secretion. There are two divisions of the autonomic nervous system I mentioned earlier, the sympathetic and parasympathetic divisions, and they do very different things. The sympathetic nervous system controls things like the stress response, so the fight or flight response that you may have heard of before. It is responsible for expending or burning off energy. The parasympathetic division works in opposition to the sympathetic nervous system. It works to conserve energy, so it governs things like homeostasis. Okay, so it brings the body back to a resting state. What's interesting is that the sympathetic division and the parasympathetic division control similar organ structures, but they do it in very different ways. When we encounter a stressful situation, the eyes, for example, you might find pupil dilation occurring so we can gather more information from our world, our visual world. The parasympathetic nervous system, on the other hand, constricts the pupil, allowing less light in. You might find that there's increased heart rate in response to a stress situation. The parasympathetic division, on the other hand, reduces heart rate. So they work in opposition with one another, but they control similar organ systems. Okay, so let's summarize this lecture for now and cover what we've discussed so far. Neurons are the basic unit of the nervous system. Glial cells provide necessary support for neurons and include cells such as astrocytes, myelinating glia, microglia, as well as radioglia. Astrocytes provide critical protection in the form of the blood-brain barrier. The nervous system can be divided into two major divisions, the central and peripheral. The central includes the brain and spinal cord, anything encased in bone, and the peripheral nervous system includes the somatic and autonomic nervous systems. The autonomic nervous system can be further subdivided into sympathetic, which governs the stress response, or the parasympathetic, which oversees homeostasis. So that's it for this lecture. Don't forget to complete your reflection.